For most people, the hepatitis B vaccine is something that happened long ago, often in infancy, and then disappeared from memory. Years later, the virus might enter the body, and nothing happens. No illness, no liver damage, no awareness the encounter even occurred. That quiet outcome raises a question. If the virus still reaches the body, what exactly did the vaccine change? To understand that, we first need to see what normally happens when the virus arrives unannounced. Hepatitis B does not behave like a sudden attacker. It behaves like a quiet guest. After entering the bloodstream, the virus travels to the liver. Not because the liver is weak, but because liver cells contain the molecular machinery the virus needs to reproduce. The virus slips into a liver cell and releases its genetic material. The cell continues living, but now it is producing viral particles. At this stage, the body often feels nothing. Damage does not begin with the virus itself. It begins when the immune system finally recognizes that infected cells exist and tries to remove them. Ironically, symptoms appear not because the virus is present, but because the immune system responds late. The infection becomes harmful when recognition comes after replication. So the key variable is not entry, it is timing. And that is precisely what the vaccine changes. The hepatitis B vaccine does not contain the virus. It contains only a single viral surface protein, a fragment the virus normally uses to attach to liver cells. By itself, the protein cannot infect, reproduce, or damage tissue. Instead, it functions as information. When this protein enters the body, immune cells treat it as a pattern. They examine it, bind to it, and begin building a catalog entry, a biological record that this structure exists. This stage is not defense, it is education. Helper immune cells activate, signaling that the pattern is meaningful. B lymphocytes refine their receptors to match the shape precisely. Some become antibody-producing cells. Others become memory cells. Nothing is being fought. The immune system is preparing a future response before the real encounter ever occurs. In other words, the vaccine moves the recognition step earlier in time. After vaccination, specialized cells remain in the body for years. They are quiet and inactive, but they are not gone. They hold a molecular memory of the viral surface, not the whole virus, only the identifying feature needed to recognize it quickly. This memory changes the sequence of events. Normally, infection follows this timeline: entry to replication, to detection, to response, to damage. After vaccination, the order shifts: entry to detection, to response, to minimal replication. The virus may still arrive, but the immune system no longer needs to discover what it is seeing. Recognition becomes immediate. And speed, more than strength, determines the outcome. Now imagine the virus entering the bloodstream years after vaccination. It reaches the liver exactly as before. It attempts to enter liver cells exactly as before. But this time, antibodies already circulate, shaped specifically to attach to the viral surface protein. Many viral particles are neutralized before entering cells. Those that do enter encounter a second defense. Memory cells activate within hours, not days. Replication never reaches the level required to trigger widespread cell destruction. From the outside, nothing happens. The virus was not prevented from arriving. It was prevented from becoming significant. Protection is achieved not by blocking exposure, but by shortening the delay between detection and response. Hepatitis B becomes dangerous when infection persists long enough to damage liver tissue repeatedly. Without prior immunity, the body needs time to learn what the virus looks like. During that learning period, viral numbers increase. With vaccination, the learning period already occurred in the past. So when exposure happens, the body acts immediately, before large-scale infection establishes itself. This is why vaccinated individuals may encounter the virus yet never develop illness. The immune system resolves the encounter before symptoms can exist. The vaccine does not create an impenetrable barrier. It creates a shorter timeline. Because the vaccine works through timing rather than exclusion, exposure can still occur. A small number of viral particles may enter cells briefly. Antibodies may not neutralize every particle instantly. But disease requires sustained replication. By interrupting replication early, the vaccine makes severe infection unlikely, rather than impossible. This distinction matters. Medicine often changes probability rather than eliminating possibility. The hepatitis B vaccine is a clear example. It does not stop encounters, it stops consequences. Many treatments aim to remove threats after they appear. Vaccination does something different. It shifts the moment of understanding earlier. Instead of reacting to damage, the body anticipates it. The immune system does not become stronger in a general sense, it becomes faster in a specific one. And in biological systems, speed often determines survival. So when nothing seems to happen after exposure, that absence is not accidental. It is the visible result of preparation performed long before the event. The vaccine did not fight the virus at the moment of infection. It changed the moment at which the body recognized it. And by changing recognition, it changed the outcome.